Hi, I'm James Rooney, and I've been working in Dr. Hasman's lab in the Department of Medicinal Chemistry and Molecular Pharmacology, where we have been investigating the effects of N-terminal methylation on substrate localization in yeast cells. N-terminal methylation is a process by which enzymes called N-terminal methyltransferases add methyl groups to the N-terminus of a protein. In humans, two genes called NTMT1 and NTMT2 encode these enzymes, and their catalytic activity is associated with cancer. This diagram demonstrates an example of an NTMT trimethylating the N-terminus of a protein. A paralog of NTMT1 and NTMT2 exists in yeast, and it is called TAE1. TAE1 preferentially recognizes and binds to a subset of substrates by recognizing the N-terminal amino acid sequence XPK, where X can be alanine, proline, or serine, P is proline, and K is lysine. This diagram shows substrates which are known to be regulated by TAA1, grouped based on their biological functions. This study investigates the possibility that if protein N-terminal methylation were prohibited in some way, the substrates may localize to a different area of the cell, thus indirectly altering the substrate's functions. This diagram shows how blocking N-terminal methylation by replacing the TAA1 gene with a gene without N-terminal transferase activity should, in theory, result in the same localization as blocking the N-terminus with green fluorescence protein, or GFP. It is important to note that both of these localizations are expected to be different than the localization of the substrate with the TAA1 gene in an open N-terminus. Using a transformation reaction, wild-type GFP strain cells containing the TAA1 gene were heat-shocked, allowing the CANM-X gene to integrate into the chromosomal DNA of the GFP cells, replacing the TAA1 gene through homologous recombination. Following the transformation reaction, transformants are grown on YPD plates containing the antibiotic G418 to select for colonies containing CANM-X. These colonies were then grown in liquid cultures, and the DNA was extracted to be amplified through a polymerase chain reaction. This specific PCR uses a negative 191 forward primer and a positive 207 reverse primer to specifically amplify the TAE1 locus. The PCR products are then compared to wild-type and TAA1 deletion standards using gel electrophoresis to confirm the presence of the CANM-X gene at the TAA1 locus. In the picture, strains containing the CANM-X gene are seen above those containing the TAA1 gene, as CANM-X is approximately 2,000 base pairs in length, while the TAA1 gene is approximately 1,100 base pairs in length. After confirming the deletion of TAA1, the samples are stained with DAPI to visualize the nucleus and loaded on an agarose gel pad between two cover slips. The localizations of the substrates can then be visualized using confocal microscopy because the substrates were tagged with GFP. The visual at the upper right shows examples of a few potential localizations of substrates in yeast cells. The panel of images below shows the localization of MRX7 in a TA1 deletion strain of yeast. MRX7 is a yeast protein that localizes to the mitochondria and associates with mitochondrial ribosomes. The localization of MRX7 in the TA1 deletion yeast strain was compared to the localization of MRX7 in the wild-type yeast strain. The localizations of many other substrates were also visualized and compared to their corresponding substrates, but the images were less clear. The localizations of at least two substrates, both with mitochondrial functions, appeared to be affected by N-terminal methylation based on confocal microscopy, but the results were inconclusive. In order to obtain more conclusive evidence, a more quantitative method of evaluating substrate localization must be pursued. Mitochondrial fractionation is one possible method that could be used to evaluate the localizations of these two substrates more quantitatively.